So Rosemary and I will tell you how we use CRISPR and Cas9 to actually probe the microRNA pathways. So that's what we were working on in the lab. So we don't work on CRISPR-Cas9, we work on other things. And in my lab in particular, we work on microRNA targeting. So how to identify microRNA targets. And you will see that this raises some very specific questions which could only be addressed by genome editing. So for us, CRISPR and Cas9 was a great help. So the main thing about microRNAs is that they are supposed to have many targets. This is a very simplified model. In fact, in mammals, microRNAs are predicted to have hundreds of targets each. So each microRNA is supposed to target hundreds of genes. And this is due to the fact that the determinant for microRNA specificity is very short. That's this segment, nucleotides 2 through 7 of the microRNA. And so this is also called the seed. So in fact, that's why people call the seed of sgRNA, because it was an analogy to what happens for microRNAs. The seed of a microRNA needs to be perfectly paired to a target for the target to be repressed. And of course, because the seed is so short, you expect to find seed matches almost everywhere. And so that's why you expect to have so many targets for each microRNA. And so that's the main issue with microRNAs. First, they have a very low specificity, so many targets. And then when you quantify the repressive effect of microRNAs on each of their targets, it's very small. So you have many targets, and each of them is hardly repressed to the point that sometimes it's even hard to detect. And so one of the main questions now is, how can we tell how microRNAs work? Through what target do they actually achieve a phenotype? And how do all these small effects sum up to perform some big biological function? And so we decided to use genome editing to address it in a very specific manner. Look, you're starting from this situation where you have a microRNA possibly targeting many genes. And using genome editing, you can actually mutate the microRNA. In this case, you would lose the interaction of the microRNA with all of its targets. So this will tell you the global phenotype, which is controlled by the microRNA through all of its targets. But of course, you can also mutate the binding site for the microRNA on one individual target. And this way, you will only disrupt the interaction between the microRNA and one given target, and you will keep all the other ones. So this will tell you what is the contribution of that particular interaction to the global phenotype. And then you can combine the two mutations, and if you were careful enough, if you took a mutation in the microRNA and a mutation in the target which are compensatory, meaning that the mutated microRNA can bind the mutated target, then in that case, you can restore specifically that particular interaction between the microRNA and that target and lose all the other interactions, okay? So that's another way, like the reciprocal experiment to this one, another way to actually assess the contribution of one interaction to the global phenotype. And so that is a strategy which is currently being developed in my lab by Isabelle Busseau. So Isabelle Busseau will give a talk later today. She's been doing that with Delphine Mazet and Alexandre Gatto, who is now in the audience. And so I won't talk so much about that because Isabelle will give you some more details later. Also in the lab, we are using that on human cells, and this is done by Gaël Dupin, who is also in the audience today. So basically, Gaël had to mutate a microRNA gene, which is miR34A, in these human cells. And so he's been using genome editing for that. So by using an sgRNA directed against the miR34A gene and providing an homology donor, which contains a neomycin resistance cassette flanked by homology arms. So if you're interested about the technical details, this is how we did. So basically, these kind of things, the resistance to antibiotics may have to be optimized from cell type to cell type. So this is for HCT116 cells. These cells are great to work with CRISPR and Cas9 because they are essentially diploid. You know that many cultured cell lines, they are very aneuploid, which means that you have many alleles for every gene, which means that if you want to mutate all of the alleles, you have to be very lucky. You have to target every allele. With HCT116, you have just 2 alleles to hit, and it seems to work very well. Perhaps there are additional things which make HCT116 very responsive to CRISPR-Cas9, but at least this works very well, and this cell line has become extremely popular since the advent of CRISPR and Cas9. So basically, that's how Gaël has been doing. He's been selecting the mutant cells with G418, and so that's the doses he's been using. And now we have isolated clones, each of them being resistant to the antibiotics, and now Gaël is analyzing them. Also in the lab, we've been also using CRISPR-Cas9 in another cell line, C2C12. This is a murine cell line, and this was done by Natalia Pinson, who used to be a postdoc in the lab. In her case, she didn't have to mutate a microRNA gene. She had to mutate the microRNA binding site in one particular target, which is called TMSB4X. So she also used CRISPR to make the break and homologous recombination to mutate the binding site. So she mutated this wild-type site into this. This is a sequence which is not bound by any murine microRNA. And in the same time, when she was mutating the binding site, she also introduced a cassette, a reporter cassette for miR1, miR206, with a luciferase reporter containing binding sites for that microRNA. This would allow us to measure the activity of miR1, miR206 in these cells. And her results were that when you mutate the binding site on TMSB4X, you're actually decreasing the expression of the reporter, meaning that the microRNA, miR1, miR206, becomes more active on the reporter. This told us that probably TMSB4X, which is a very abundant mRNA in these cells, probably it's so abundant that it can sponge the microRNA to the point that when you remove the binding site on that gene, you're actually releasing some microRNA, which can then better target the other genes, including the reporter gene. So that's the way she has been using CRISPR and Cas9 to mutate a binding site for a microRNA. Okay, so I think I'm running late, so maybe it's your turn, Rosemary.
So I'll follow on from Hervé, where we also use CRISPR-Cas9 to probe the microRNA pathway, and we also wanted to delete a particular microRNA in the genome, microRNA 3173. Why did we want to do that? Because we've developed a model based on a lot of experiments using a lot of different techniques that basically show a novel mechanism of regulation of the protein DISA. DISA, you may know, is a major enzyme in the RNAi pathway, and it's also a tumor suppressor. It's a very important protein in the cell. And we found that the expression of DISA protein is dependent on the presence of a particular microRNA, microRNA 3173, that's found in the very first intron of the pre-messenger RNA of DISA. And why is this important? It turns out that depending on the presence of certain factors, for example, microprocessor that is going to cleave this microRNA, that will lead to induction of a low expression of DISA protein because we cleave the pre-messenger RNA. It releases this microRNA, leading to a higher level of microRNA expression in the cells. On the other hand, we identified a factor that actually competes. It's known to compete with microprocessor, and indeed it does compete with microprocessor for the processing of this microRNA. So if this factor is abundant, we actually induce a different pathway that leads to high expression of DISA, which is good for cells, and low expression of the microRNA. This becomes a little bit more complicated because it turns out that this particular microRNA targets this factor here. So we end up with, we develop these amplification loops that either favor this pathway. If this factor is low in the cells, we favor the processing of this microRNA, low DISA expression, high expression of the micro, which then induces this amplification loop, which is negative for DISA expression. On the other hand, if this factor is abundant, we induce an amplification loop that promotes high DISA expression and controlled growth of the cells. So this is a fairly complex model, and we really wanted to prove our model. And as you can see, the presence of this microRNA, the physical presence of this stem loop in the intron, is really central to this model. So to prove our model, we wanted to delete this microRNA. And of course, how to delete this microRNA? There's different possibilities. We could have used sort of a plasmid-based exogenous approach, but we really needed to keep the genomic context. And this particular intron is 25 kilobases long, so I think that rules out plasmid-based approaches. RNAi, as Hervé mentioned, isn't appropriate for this approach either because the microRNA is in the intron of the pre-messenger RNA, which is 70 kilobases long. It's also part of the pre-messenger RNA molecule. So it would be impossible with RNAi to specifically remove the microRNA without affecting the DISA messenger. So RNAi wouldn't be appropriate either. And I forgot to mention, we could have possibly used mouse knockouts of this microRNA, but the problem here is that this microRNA is specific for primates. It's not expressed in mice. So mouse approaches weren't possible either. So it seemed like the only possibility was CRISPR-Cas9. So we are not expert in CRISPR-Cas9. We went about this. We did actually use web-based prediction tools. We used ChopChop, which is developed by the Broad Institute, and I think quite a reputable program. So, and it's very simple to use, and we told ChopChop that we wanted to make a deletion in this region, and we picked the top hit, which has the least number of off-targets, and we then cloned this sgRNA that ChopChop gave us into a plasmid, very standard CRISPR plasmid that we obtained from Adgene, and that comes from the Zhang lab. Now, the cell line, the cells that we wanted to use are very special cells. They are derived from a patient, an ovarian cancer patient, so they're patient-derived xenograft cells. So they're very difficult to transfect. And indeed, we had a very low rate of transfection using lipofectamine 3000. So we decided to sort the GFP-positive cells by FACS. So we're selecting only the cells that express the CRISPR-Cas9. And then we subjected them to limited dilution cell cloning. Once again, we came across a little technical difficulty because these cells were normally in limited dilution cell cloning. You dilute the cells that there's one cell per well. But the problem is these cells really don't like to be lonely. They die if there's one cell per well. So we had to adapt the protocol a little bit. So we ended up seeding the cells very sparsely into pretty large dishes and letting the cells grow into small colonies that we were able to then remove using cell cloning discs to individual wells. And that seemed to get around this little technical problem that we encountered here. So if you're using cells that are a bit tricky like that, you might want to consider that. Then when we let the cells grow up after about 1 month of cell growth, we screened them by fairly standard PCR-based genotyping. And we obtained, out of the first 14 clones that grew up, we obtained a clone that seemed suitable to us. I should mention that we didn't use homology-directed deletion. We just took advantage of the non-homology and joining pathways, so we were just looking for random mutations, deletions. And this is the clone that seems suitable to us. We have a small deletion in the middle of this microRNA. And when we measure the amount of the microRNA produced in the cells, actually we totally lose the expression of this particular microRNA. So then we thought we had our cells. So then I just would like to show you what we then did with these cells. So to go back to the original model we had, we wanted to show in these cells that we had lost the ability of this factor ILF3 to regulate the expression of DICER. And indeed, that's what we got. If we look at the wild-type cells, when we knock down this factor, we also lose expression of DICER. Whereas in the over to deleted cells, so remember we've only deleted 30 nucleotides in these cells, when we knock down this factor, DICER is no longer subject to expression, changes in expression. So that helps to prove our model. Again, if we look at the proliferation of these ovarian cancer cells, we see that when we overexpress ILF3, that slows down the growth of these cells, which is good because they're an ovarian cancer cell line. Whereas when we've deleted the region corresponding to the microRNA, we lose the ability of this factor to control DICER expression. So that helps us prove the model that we developed using many other techniques. Do I have 1 minute or? Well, okay. I'll just switch gears a little bit and show you something else we've done with CRISPR in the lab that helped us once again to get over a little technical hitch that we've encountered. So this is a different issue. So now, this time we're using CRISPR to tag endogenous proteins to do interactome studies. So interactome studies, they're fairly typically done using a very well-described method from the Nakatani lab, where cells are infected with a retrovirus expressing your cDNA of interest that's got a flag. We use flag HA tags, generate stable cell lines, do a dignum extraction, and do very nice tandem affinity purification, which allows us to do very clean mass spectrometry, which is great, except there's actually a technical limit here because there's a size limit on the cDNA that you can clone into the lentiviral vector. And that means that there's a lot of proteins actually that you can't study using this technique. And CRISPR, we realized that CRISPR could help us get around this. So instead of using the lentiviral vector, we now use CRISPR to tag the endogenous protein. So here, this time, we use homologous recombination. So we have the CRISPR plasmid, and we transfect in the cells an ssODN, which contains the tags plus homology arms that are going to direct that tag to the either the N-terminus or the C-terminus of the protein of interest. And that's actually helped us get around this technical problem and allowed us, I'm just showing you one example here, but we've done it for many proteins in the lab now. This is a protein that's pretty big. It's 350 kilodaltons, so it's getting up to be pretty large. And this actually, before we could not possibly have done a mass spectrometry of this protein. And now with CRISPR, actually, we've been able to get interactome studies of many proteins like this. So thank you very much. Just like to mention people in the lab, particularly Marion Asmortel, who in joining the lab brought with her a lot of experience and competence in CRISPR, which was great. Xavier's also been very involved in the development of the tagging of the endogenous proteins for the mass spec studies. And Jérôme and Kader and Marina and Pierre have also been involved in this work as well. Thank you.